thank you for that introduction. I've been asked to talk a little bit about my experiences with the fellowship, some of the opportunities that I've been presented with, as well as provide some information for laboratory directors or as well as some university scientists as well that they could benefit and how they can benefit from this program. Sorry, I'm a little short. So yes, I've been given about 10 minutes to talk about my 2 years as a fellow, so we'll see how this goes. Just a brief overview on what the fellowship is. It's a 2-year postdoctoral fellowship that the PCC provides support. They give a competitive stipend. If you've been a postdoc, you understand that, at least compared with NIH standards. They also give the potential for a 3rd year of PCC support following the 2 years. It's up to both the fellow, the PCC, as well as the laboratory directors. The first 3 months is designed to support existing lab research and to learn about the intricacies of sample testing in a WADA-Accredited lab. And then after 3 months, a fellow can work in a different lab with PCC approval, some academic institutions. So we've had some scientists talk here today from academic universities, and they could certainly benefit from this as well with after a fellow gains some knowledge in a WADA-accredited lab, can move on to a university to do research. This is very important. The next part, there's a research focus, but you're supposed to familiarize yourself with laboratory procedures. It's a very broad term. If you're unfamiliar with what goes on in a laboratory, whether you're a policy administrator or if you've never visited a lab, I don't know that you'd understand all the intricacies that do go on in a laboratory. It's not just send us a sample, we drop a piece of, drop a urine into a machine and call it good, call it what it is. There's many steps involved in this process, many places where optimization can occur. So it's very important to familiarize with yourself with these procedures. And again, another important thing in terms of familiarizing yourself is anti-doping, as we've learned today. It's a very small field, so it takes quite a while to be able to understand where the holes are in the procedures or what needs improvement before you can actually make improvement. If you're a scientist and you moved into a new field, it takes a long time to learn that field before you can help to improve that field. So this is what this 3 months and then even further in a WADA-accredited lab can help do. Some of the research projects I've worked with Smyrtle. We started with a biological passport project looking at hydration markers. This was what the fellowship was granted for, and we did get a WADA grant for this. Since then, we've done an autologous blood transfusion study. So, Dr. Harrison, we do have some blood cells for you if you'd like to analyze them from a administration or a autologous blood transfusion administration study. This was PCC funded project, and it's given us an opportunity to collaborate with some medical doctors at Harvard University. Finally, another biological passport related project is we've looked at Ironman events and how athletes respond following these events. So these are, full day, 10, 11, 12 hours, however long it takes to complete an Ironman. This is very physiologically taxing, and what we've seen is in the weeks that follow, the blood profile may look a little bit different than what we would see throughout the course of, an out of competition schedule for some of these athletes. This has also given us a couple other opportunities to collaborate with an administrator. So we've worked with the Ironman anti-doping program to work with them on this project, as well as the Australian Institute of Sport, so some international collaborations as well. Most of my work has been focused in the blood doping and the biological passport area. We've also done some other projects, which I've termed other doping here. We did an intranasal testosterone administration study that was funded by the PCC. Some small molecule metabolism using in vitro methods, which Dr. Van Enu talked about a little bit today. Also PCC and USADA funded. And so we've done that twice, once with small molecules, once with peptides. And then one of the projects we're currently working on is administration of clomiphene. This is an ongoing study, also funded by the PCC. And what we're doing here, just if you're curious, so I can provide some pictures up here instead of just words. We're running a 30-day clomiphene administration in males. The reason we're providing a female fertility drug to males is that it is known to increase testosterone, and what we've seen is it certainly does that. And a lot of these subjects that we're using, these are active subjects and not just sedentary. We've seen in the first couple weeks almost doubling of serum testosterone levels, which is quite striking. But the reason that we're looking at this is to try to develop a detection window. If you're familiar with this drug at all, it undergoes the pharmacokinetics of it. It doesn't clear from the body very quickly. It undergoes enterohepatic recycling, which means the liver can kick it out into the intestine. It goes back into the portal, kicks back into the liver, keeps cycling and cycling and cycling. It takes a very long time to clear from the body. So what we're trying to do is both establish detection windows for urine and blood for this compound to kind of understand maybe when an athlete has taken this, if we find positive tests for it, and what we can, how we can interpret the data that we may find in the laboratory. A couple other things briefly. What this fellowship has allowed is me to understand a lot of the behind-the-scenes work that goes with the research projects. A number of those projects up there, we had to use human studies for administration, because that does provide the best type of data that we can get is when we get them from humans. The medical and ethical approval for research studies, that takes quite a long time to do. What I've learned is from project inception until we finally get approval to start the project, it takes probably 4 to 6 months. So you like to move a little bit faster, but research does sometimes take time when you want to do it properly and when you have to go through all the channels that you need to do in order to use humans or use people for as subjects for the research studies. We've had a close collaboration with the University of Utah, so it's allowed some academic collaboration with our WADA-Accredited laboratory at Smyrtle. It's taught also a lot about sample collection and organization, so I got certified as a phlebotomist because it allows a lot quicker analysis, what we can do blood draws in our laboratory and for the university as well. If we want to run quick studies or as a part of this research studies, the research studies that we run, it gives us a little bit easier collection methods to do this. And finally, it also involves collaboration with a lot of members of the medical field. All of these studies, especially when we're doing drug administration studies, you have to have a medical doctor on board with you to examine side effects to make sure that the health of the subject or the health of the patient is of utmost priority. So it's involved a lot of different talk with medical members, so understanding how things work in the medical field. Conferences like this, you also get to understand how things work for administrators, for lawyers, everything. So it gives this fellowship has provided a pretty broad knowledge base for future in anti-doping. Some other opportunities. I've had a freedom to work in a lot of different areas. As I showed you, I broke it down into 2 subjects, the blood passport and the other doping section. Daniel Eichner has given me a lot of freedom to work in different areas of the laboratory to try to find what interests me for both a research standpoint and for understanding testing procedures, which part of the lab would I like to work with testing procedures, where do we need help, things along those lines. It's provided a lot of scientific meeting invitations, which include the PCC meeting. I was here in 2015, and it's great to be back again. The Cologne workshop, as well as the USADA science meeting. These are all great opportunities with all different focuses as well. This meeting provides a different scope than the Cologne workshop, which is very laboratory-based, and then the USADA annual science meeting as well, which is a lot of basic science, which is very important for creating new research projects and understanding the background to a lot of the work that we do. Other laboratory tests, finally, I've kind of migrated toward the EPO analysis, growth hormone analysis, as well as some APMU work. Smyrtle's the only laboratory in the United States that does the blood passport testing, so we're the only one with an APMU, so that's provided a great opportunity to understand these processes and to work in that area. Finally, some information for laboratory directors. So the institution, your WADA-accredited laboratory, your institution is paid overhead in addition to the 2-year stipend. So not only do you get, it's not a free employee, but a near free employee, also a postdoc. Some funding is provided to the laboratories to support curriculum development and some time mentoring as well. And what I've been told from Daniel is what they look for is a quality fellow versus a quality project. I mentioned earlier, if you're just joining the anti-doping field, it's very difficult to come up with a meaningful project from the start. If you're unfamiliar with where the holes are or with how the anti-doping system works or with current research that's going on, that's going to take a little bit of time to get used to and to understand. So for the laboratory director, I've been told to have you look for a quality fellow as opposed to necessarily a quality project or a quality proposal. Some laboratory commitments. The laboratory is committed to developing long-term talent for the field of anti-doping, as well as to provide a plan to the PCC detailing how the fellow will be supported and mentored. So if you're going to use a fellow in the laboratory, maybe which portions of the laboratory, what you're going to have them do in the laboratory, and how what they're doing is going to help with the research from the lab as well, and how it's going to work for the PCC. So finally, and then I'll let Li Ying talk up here as well, it provides a great introduction to the anti-doping field. So it has been 2 years. You learn quite a bit in 2 years, and I think it's very surprising to myself where I was 2 years ago and kind of what I've been able to learn since then. It's beneficial to fellow, gives you a lot of opportunities for growth and for productivity. It's beneficial for the laboratory. You get a near full-time research staffer that works in the laboratory, and then once they learn the intricacies of the lab, they can provide some details on method improvement or some optimization of methods as well. And then finally, we hope to be, we hope to provide great insight to the field of anti-doping science. You know, it is a very difficult task, but that's kind of what we try to do, is to provide good research and good data for the field of anti-doping science.